the microscopic alchemist. If you look closely at a patch of powdery mildew on a zucchini leaf, you see a graveyard in the making, or rather a battlefield. To the naked eye, it is just an unsightly white dust, a sterile velvety coating choking the life out of the plant. But if we zoom in, scaling down our perspective by a factor of 10,000, a terrifying and beautiful alien landscape emerges. Here, the white dust transforms into a dense, tangled jungle of fungal threads called hyphae. They are parasitic pipelines, drilling into the plant's cells to suck out its sugars. For most creatures, this environment is toxic, a suffocating web of fungal defense mechanisms and decaying plant tissue. But for our protagonist, this microscopic wasteland is a buffet. How does a creature less than 4 millimeters long navigate, consume, and neutralize a fungal pathogen without being consumed itself? The answer lies in a brilliant combination of mechanical precision and specialized biochemistry. This is not just eating, it is a high-stakes chemical extraction. Step 1. The mechanical breaker. Before any chemical magic can happen, the subject must solve a physical problem. Powdery mildew isn't just loose powder, it is a matrix of microscopic chains. The fungus reproduces by budding off clonal spores called conidia, which stack on top of each other like fragile towers of ceramic cups. Our subject approaches these towers not with brute force, but with highly adapted anatomical tools. The mouth parts are a marvel of microengineering. Unlike predatory beetles that possess massive, jagged mandibles designed to crush and tear thick insect chitin, this creature's mouth parts are modified into delicate, specialized scrapers and scoops. Watch as it lowers its head. The mandibles move with rhythmic, hypnotic efficiency. They act like a combination of a razor-sharp paint scraper and a vacuum nozzle. With microscopic precision, they shave the fungal hyphae right off the epidermis of the leaf. It doesn't pierce the leaf itself. Its touch is so refined that it skims the parasitic fungus off the plant's skin without drawing a single drop of plant sap. The movement is systematic, clearing perfect green paths through the white wilderness like a tiny lawnmower cutting through a field of frosted glass. Step 2. Breaking the chitin. While once the fungal material is swept into the mouth, the true crisis begins. Fungi are not plants, and they are certainly not soft tissue. Their cell walls are built from chitin, the exact same tough, fibrous polymer that makes up the hard exoskeletons of crabs, lobsters, and indeed, insects themselves. Swallowing powdery mildew is equivalent to a human trying to digest shards of fiberglass or sheets of plastic. Without a way to dissolve this outer armor, the spores would pass through the digestive tract completely intact, rendering the meal useless and potentially causing internal tearing. To solve this, the subject deploys an internal chemical weapon. Chitinase. As soon as the fungal spores enter the foregut, they are bathed in a highly concentrated, specialized enzyme cocktail. Think of chitinase as a pair of molecular wire cutters. The long, tightly woven chains of chitin are held together by incredibly strong chemical bonds. The chitinase enzymes lock onto these bonds and systematically snip them apart, converting the tough, crystalline armor into simple, soluble sugars like N-acetylglucosamine. In a matter of minutes, what was a resilient protective shell becomes a soft, digestible slurry. It is a profound evolutionary irony. The insect uses a chemical specifically designed to break down insect-like armor, turned against a fungus to sustain its own life. Step 3. The neutralization of mycotoxins. But the chemical battlefield runs deeper. Fungi are notorious masters of chemical warfare. Powdery mildew doesn't just sit there and allow itself to be digested. It is packed with metabolic byproducts, secondary metabolites, and mycotoxins designed to deter herbivores. These compounds can disrupt cellular function, poison vital organs, and cause severe oxidative stress. If you or I were to ingest powdery mildew at this scale, the toxic load would trigger an immediate, violent immune response. How does our subject survive this biochemical assault? The secret lies in its midgut, a highly advanced biodetoxification lab. Here, the insect utilizes a massive army of enzymes known as cytochrome P450 monooxygenases. Imagine a high-security hazardous waste processing plant. When a toxic fungal molecule enters the midgut, these P450 enzymes immediately intercept it. They perform a process called hydroxylation, essentially attaching a highly reactive oxygen molecule to the toxin. This single chemical modification alters the structure of the poison, rendering it completely inert. It changes a deadly molecular key so that it no longer fits into any cellular locks. Simultaneously, another enzyme group, the glutathione S transferases, GSDs, steps in. They grab these neutralized toxins and attach a sugar molecule to them, making them highly water-soluble. This ensures that the dangerous compounds can be safely and rapidly flushed out of the body before they can accumulate. The ultimate energy return because it can crack the chitin shell and neutralize the fungal defenses. The subject gains access to a treasure trove of pristine nutrition locked inside the mildew. Fungal spores are incredibly rich in lipids, glycogen, and essential amino acids by burning almost zero energy on chasing fast-moving prey and instead investing its evolutionary capital into this internal chemical refinery. The subject achieves an astonishingly high metabolic efficiency. Every scrape of its mandibles converts a destructive plant plague into pure, clean energy. As the sun begins to dip lower, casting long shadows across the leaf, the relentless scraping continues. The green patch grows larger, the white chokehold of the mildew retreats. What appeared to be a simple insect grazing on a leaf is revealed to be an incredibly sophisticated biochemical engine, running a masterclass in ecological purification. But this microscopic victory is just one piece of a much larger, shifting puzzle. Because out here in the open garden, chemistry isn't the only law. Survival requires strategy, defense, and an uncanny relationship with the surrounding ecosystem, and the micro-world is about to get much more dangerous. The secret weapon of 1847 for thousands of years, humanity viewed the microscopic alchemist not with awe but with absolute blindness. We stared at our ruined crops, watched our vineyards wither into ash, and blamed the wrath of the gods, the alignment of the stars, or the bad air of a changing season. We did not know that a silent, yellow and black guardian was offering its services for free, right beneath our eyes. To understand how this creature collided with human history, we have to travel back to the mid-19th century, a time of sheer agricultural terror. The stage is Western Europe, specifically the sun-drenched hills of France and the mist-heavy valleys of England. The stakes? Total economic collapse and starvation. The White Plague arrives in the late 1840s. An invisible killer breached the borders of Europe. It arrived quietly, hitching a ride on imported American vines. Within months, a catastrophic disease swept through the vineyards of the Old World. Oidium tocheri, known to horrified farmers as powdery mildew, it was a botanical wildfire. Vines that had produced world-class wine for generations were suddenly choked by a ghostly, suffocating white shroud. The leaves curled and died. The grapes shriveled into hard, bitter marbles covered in ash. In France alone, wine production plummeted by over 75% in a matter of years. Multi-generational families were ruined overnight. Riots broke out in rural villages. The literal spirit of European agriculture was facing extinction. Enter our human protagonist, a brilliant, stubborn, and largely forgotten French entomologist and botanist named Jean-Pierre Etienne. While most desperate scientists of the era were locking themselves in pristine laboratories, mixing volatile concoctions of sulfur, copper, and vinegar to douse the dying crops, Etienne took a different approach. He went to the graveyard itself. He knelt in the dirt of a dying vineyard in the south of France, peering through a crude brass pocket microscope at a heavily infected leaf. What he saw shocked him. Amidst the terrifying white jungle of fungal threads, there was no sign of plant life. But there was movement. The discovery in the dust Etienne watched as a tiny, dome-shaped insect, vibrant yellow with stark black spots, marched directly into the center of the fungal infestation. It was a ladybug, but not the famous red and black predator that hunts aphids. This was Silobora vigentidua punctata, the 22-spot ladybird. To Etienne's astonishment, this beetle wasn't hunting other insects. It was lowering its head and methodically devouring the White Plague. It was a moment of profound revelation. In the middle of an agricultural apocalypse, nature had already deployed its own elite counter-terrorism unit, but Etienne's discovery was met with immediate, mocking skepticism by the scientific establishment. The mid-1800s was the dawn of the chemical age. The Industrial Revolution had taught humanity that the answer to every problem lay in a factory, a smokestack, or a synthetic spray. The idea that a tiny 4mm beetle could save the agricultural economy of Europe was treated as a fairy tale. "You expect us to fight a continental plague with a handful of yellow bugs?" wrote one prominent Parisian critic. Undeterred, Etienne escalated the stakes. He initiated what would become one of the earliest undocumented experiments in biological control. The high-stakes gamble. In the summer of 1851, Etienne selected 2 identical plots of heavily infected vines on a hillside near Montpellier. Plot A would be treated with the cutting-edge chemical sprays of the day, harsh lime-sulfur mixtures that blackened the soil and burned the hands of the laborers. Plot B would be left entirely alone, save for one intervention. Etienne and his assistants spent weeks collecting thousands of 22-spot ladybirds from the wild, carefully transferring them to the diseased leaves. It was a grueling, maddening task. The beetles were fragile, and many flew away. Meanwhile, the owners of the vineyard watched in horror, convinced Etienne was actively destroying what little crop they had left. The pressure was suffocating. If the experiment failed, Etienne would face public disgrace and financial ruin, having wasted a crucial growing season during a national emergency. For weeks, the chemicals of Plot A seemed to win. The mildew turned gray and died, but the victory was hollow. The harsh sulfur also scorched the delicate plant tissues, preventing the vines from producing healthy fruit. Then came the turning point. In Plot B, the fungal jungle began to recede. But it wasn't turning black from poison. It was simply disappearing, leaving behind clean, vibrant green tissue. The yellow beetles had multiplied. Their microscopic larvae, voracious pale yellow monsters, had joined the fray, scraping the mildew clean with industrial efficiency. By late autumn, Plot B produced a modest yet healthy harvest of unblemished grapes. Plot A produced nothing but withered vines and toxic soil. The tragedy of timing Etienne had proven a revolutionary truth. Humanity did not need to wage a scorched earth chemical war against nature. We simply needed to understand and amplify the alliances that already existed. Yet, history is a fickle narrator. Just as Etienne was preparing to publish his definitive treatise on using the 22-spot ladybird as a widespread agricultural shield, the French chemist Henri Marès successfully stabilized a cheaper, easily mass-produced dry sulfur dust. It required no careful handling of live insects. It could be thrown into a machine and blasted across thousands of acres in a single afternoon. The industrial machine won. Etienne's delicate, ecological approach was swept aside by the tide of chemical agriculture. His notes were archived, his yellow and black beetles, forgotten by all but a few dedicated naturalists. As the world entered a century-long romance with synthetic pesticides, a path that would eventually lead to silent springs and poisoned ecosystems. But history runs in cycles. Today, as powdery mildew develops terrifying resistance to our strongest chemical sprays, modern scientists are returning to those dusty archives. They are looking back at the forgotten drama of 1851, realizing that Etienne wasn't wrong. He was just a century ahead of his time. The secret weapon of the past is fast becoming our brightest hope for the future. The modern frontier and the mirror of hubris today, we find ourselves standing on the edge of a new agricultural precipice. The industrial chemical warfare that began in the 19th century has reached its logical, devastating conclusion. Across the globe, powdery mildew has done what life always does. It adapted. The fungus has mutated, developing a terrifying resistance to our most advanced synthetic fungicides. We spray harder, mix more toxic chemical cocktails, and yet the white shroud keeps creeping back, threatening our global food security in an era already strained by a changing climate. But inside the ultra-modern greenhouse complexes of the 21st century, monoliths of glass and automated hydroponics, a quiet revolution is taking place. We are finally learning to stop fighting the ecosystem and start copying it. Automated ecology step inside a high-tech vertical farm today, and you might expect to see nothing but cold steel, LED arrays, and sterile white walls. But look closer at the leaves of the crops, and you will see a familiar sight. The 22-spot ladybird has traded the sun-drenched hills of 19th century France for the climate-controlled racks of modern agritech. In the field of integrated pest management, this beetle is no longer a forgotten historical footnote. It is a precision asset. Modern drone technology and artificial intelligence are now being designed to mimic what this insect does naturally. Automated cameras scan crops at the millimeter scale, using hyperspectral imaging to detect the earliest chemical signals of a powdery mildew infection long before it is visible to the human eye. Once identified, instead of blanketing the entire facility in chemical poison, we deploy the microalchemists. We are even studying the beetle's internal biochemistry to revolutionize human medicine and materials science. The chitinase enzymes that the insect uses to liquefy the fungal armor are being analyzed by bioengineers. We are attempting to synthesize these exact molecular wire cutters to create new eco-friendly antifungal medications for humans, as well as biodegradable plastics that can break down without leaving toxic microplastics in our oceans. The microscopic wasteland has become our blueprint for sustainability. The mirror of the micro world there is a profound, almost humbling irony here. For centuries, humanity defined progress by our ability to dominate, pave over, and chemically sterilize the natural world. We built towering factories and synthesized complex molecules, convinced that human ingenuity could outsmart billions of years of evolutionary trial and error. We looked at a 4 millimeter beetle eating dust on a zucchini leaf and saw something insignificant, a primitive creature easily bypassed by human progress. Yet, as our modern chemical solutions fail us, we are forced to bend our knee to the wisdom of the leaf. We are realizing that the ultimate pinnacle of technology is not a louder machine or a more toxic spray. It is the elegant, waste-free, hyper-targeted biochemistry of a ladybug. The beetle doesn't create pollution. It doesn't leave behind a toxic legacy. It takes a destructive plague and transforms it into life, leaving the environment cleaner than it found it. This realization carries a heavy, cautionary truth. It forces us to confront our own cosmic fragility. We pride ourselves on being the masters of this planet, capable of splitting the atom and launching vessels into the void of space. But our dominance is an illusion built on a fragile foundation. If the microscopic relationships that sustain our soil and our crops were to collapse, if the silent guardians like the 22-spot ladybird were to disappear, our grand civilizations would wither just like the French vineyards of 1847. A quiet simp, pony of survival, we share this floating blue marble with 1 trillion tiny engineers, each running a complex biochemical empire of its own. They do not need us. The mildew will find a way to grow, and the beetle will find a way to scrape it clean, with or without our permission. It is we who need them. The next time you walk through a garden or pass by an overgrown patch of weeds at the edge of a concrete sidewalk, pause and look closer. Do not just see weeds and do not just see pests. Look for that tiny flash of brilliant yellow and stark black, moving with rhythmic, ancient precision across a dusty leaf. Listen closely, past the hum of traffic and the noise of the human world, to the silent, invisible war being waged in the undergrowth. It is a masterpiece of microengineering, a high-stakes chemical extraction, and a timeless reminder that true power does not roar. It scrapes, it neutralizes, it survives.